Welcome students to this new series on medicinal plants. In this series, we will explore the fascinating world of natural remedies and the powerful healing properties of plants. In this video, I will introduce you to the vocabulary of phytotherapy, as well as the art of harvesting plants. You will learn the different parts of the plants that are used for medicinal purposes, as well as the best time and methods for harvesting them. Are you ready? Let's begin. Hi, I am Tommy. I'm a human nutrition student and researcher at the university. What I do in this type of videos is to synthesize my classes at the university, so then I can explain and teach you the best and most valuable information you need to know. So let's begin this video by talking about the introduction to the phytotherapy, the vocabulary. So let's get started. Introduction. The definition of phytotherapy is basically using medicines derived from plants or herbs to treat or prevent health conditions. Indeed, the etymology and etiology are from Python, which means plant, and therapeia, which means healing or medical treatment. It's a complementary medicine practice. And a medicinal plant is a plant used for medicinal purposes. A drug is a substance that has physiological effect when ingested or otherwise introduced into the body. It's the part of the plant that contains the active principle, for example, the leaf, the root, the bark, the flower, whereas medicinal drug is not the same. This is referred to the active principle alone. A medicinal drug is the active principle of pharmaceutical drugs. Drugs are referred to the plants that contain those active principles. Vegetable preparation, drug administration through herbal teas, extracts, oils, macerations. We will have a video talking about that in detail. Traditional medicinal plants are plants used for medicinal purposes for generations and are often part of traditional medical systems. Active principle. It's the chemical compound that is responsible for the physiological effects of that drug. And phytotherapeutic products, they are more regulated and available by prescription in some other countries, particularly here in Europe. There is a controversy surrounding regulation of dietary supplements, and some argue that the current regulations are not strict enough to ensure the safety and efficacy of these products, because many manufacturers, what they do is to label their products as dietary supplements instead of pharmaceutical drugs, because they follow less strict regulations indeed. And these phytotherapeutic products are sold in the US over the counter as dietary supplements, as you can see. They have not undergone rigorous scientific studies of effectiveness, safety, or quality. So the legislation behind the medicinal plants, as you can see, varies by each country. Here in Europe, we tend to be more strict than in the US. So now we talk about the collection, harvesting, preservation, and also storage of medicinal plants. So from phytotherapy, we have the identification of medicinal plants, then the collection of them, preparation of plant material, extraction of active principles. Then we have isolation and purification stages. Then a formulation of phytotherapeutic products, clinical trials to actually check them, regulatory approval, and then marketing and distribution of them. So, how to harvest and process medicinal plants? Types of plants we can find: wild or cultivated, autochthonous or alloctonous, acclimatized or exotic. Acclimatized means that they have been imported and they got used to the weather. So, wild plants and cultivated plants, the main differences. In wild plants, the inconvenience are the lower production, of course, irregular growth, geographical dispersion, variable active ingredient content. The active principle is in who knows what amounts, expensive collection, risk of contamination because it's out there, wild, and risk of extinction of that species as well. It's only recommended when there is an abundant natural production, it's cost effective, the harvesting, when the cultivation is not available, and when there is very low demand. On the other hand, we have the cultivated plants. The benefits are abundant and good quality harvests, similar growth between them, selection techniques that ensure a higher drug quality. We also have a controlled active ingredient quantity. The active principle is actually quantifiable. Localized production, very important, and higher control and lower manipulations. The convenience are market saturation due to overproduction of it and more fragile compared to the wild plants. So from a wild plant to a cultivated plant, we have the domestication of the plant. And what are the steps? The first one is to study the natural habit. Then to collect that plant and the seeds. Look for a genetic improvement. Number 4 is conditions, possible problems and duration of the cultivation. So actually create an analysis behind it. And number 5 is the economic evaluation. So to propagate the new species, we can do it via sexual form using seeds or asexual form using cell and tissue cultures. So callus cultures are undifferentiated cells together with auxins and cytokines. Then we have organogenic cultures. This is when there is a lot of active principle in specific organs of the plant. And we also have transformed hairy root cultures. And this has to do with the infection by Rhizobium rhizogenes bacteria. And to do this, to promote the domestication of the plant, we use vegetable biotechnology. This is, for example, the use of elicitors. These are substances that are used by the plant in a given situation, for example, when it's attacked. And the use of metabolic engineering will force the production of those compounds of interest, those elicitors, when we want. But it's very difficult to replicate indeed. Now, taking a look in more detail at drug harvesting, what are the factors? The plant age, the season of the year, also the time of the day. These are factors that will determine the composition of that drug. Remember, drug is the plant, the part of the plant with the active principle. Post-harvest alterations. Yeah, because we have enzymatic reactions and external alterations that will occur after we harvest them. What's the solution? Enzymatic inhibition, which is the dehydration of the plant, for example, and enzymatic inactivation, the destruction. Inhibition types, we have natural drying, such as sun and shade, artificial drying using drying tunnels, infrared or microwave, or even freeze drying at -40, -80 degrees Celsius, and then sublimate. On the other hand, to destroy those enzymes, we can use boiling alcohols or liquid vapors, such as water, alcoholic, or dry heat. So what are the parts of the plant that will have the medicinal purposes? The leaf, and we have to harvest it when it's green. The flowers in the pre-flowering period, because this corresponds to the puberty of the plant, and during puberty we have more hormones, so more active ingredients. Then fruits and seeds as well, when ripe only. The bark in vegetative processes, this is in spring and summer. And the root and rhizome, not in vegetative processes, so in autumn and winter. Then once we harvest the plant, the drug, we have to store it in a cool, dry place, out of light, isolate from the atmosphere, in a non-porous container, and to avoid the development of parasites and microorganisms. Control the storage time as well. This is all done to prevent the post-harvest alteration. And last but not least, we have to do a quality control. This is for reproducibility and protection. Many factors are involved: finding the identity of the plant, the state of preservation of it, the adulteration, if any, the amount of active ingredient, we have to quantify it, and also post-harvest processes. What are the elements of quality control? Clear and scientific definition of the plant with its taxonomy, identity. So what are the characteristics of that plant? Evaluation. So the quantity of active ingredient, preservation and stability, and the purity. So the humidity, the hash, and the contaminants it may have. To do this quality control, we use chromatographs for the analysis. And the control is at all stages. So all stages, as you can see here, from the identification to the marketing and distribution. Okay, this is it for this video. I hope you understood everything I said. If you didn't, there is a comment section down below. Remember to like, subscribe, and turn on the notifications so you get notified every time I upload a new video. If you find value in this, please share it to your friends and family to spread this knowledge to them as well. Thank you so much for watching, and I will see you in the next one, in which we're going to talk about the active ingredients of medicinal plants and the phytotherapeutic preparations. I will see you there. Hope you enjoyed.